Hello friends, today we shall discuss about genetic disorders. Now, genetic disorders are broadly classified into two categories, Mendelian disorders and chromosomal disorders. Now, Mendelian disorders are caused due to defect or mutation in the individual genes, whereas the chromosomal disorders are caused due to change in chromosome structure and number. Now, Mendelian disorders are further classified into allosomal allosomal and autosomal disorders. Now, allosomal disorders are caused due to mutation in the genes located on allosomes or sex chromosomes, whereas autosomal disorders are caused due to mutation in the genes which are located on autosomes. Now, autosomal and allosomal disorders are further classified into dominant and recessive disorders. Now, dominant disorders are caused when the dominant gene becomes defective. Recessive disorders are caused when the recessive genes become defective. Example for autosomal dominant disorder is myotonic dystrophy. For autosomal recessive disorder, the examples are sickle cell anemia and thalassemia. For autosomal diseases, the example is Rett syndrome. It is not there in syllabus. Okay. And for allosomal recessive diseases, examples are hemophilia and color blindness that we have already discussed. Okay, so we are not going to discuss about allosomal recessive or sex-linked recessive disease again in this part, in this clip. Now, first we'll start with Mendelian disorders. Now, Mendelian disorders are caused due to alteration or mutation in the single gene, not the entire chromosome, just single gene. Examples: hemophilia, color blindness, sickle cell anemia, phenylketonuria, thalassemia, etc. In sickle cell anemia, the shape of the RBCs turn into sickle shape when the oxygen levels are low. The normal shaped RBCs, they are biconcave. Here you can differentiate between the normal RBCs and the sickle shaped RBCs. Now, sickle shaped RBCs, they block the blood vessels and carry little or no oxygen at all. Thus, it affects the normal functioning of the hemoglobin. Now, let's see why this change in shape of RBCs occur. Hemoglobin is made up of 4 polypeptide chains, 2 alpha chains and 2 beta chains. In the beta chain, okay, you can see, in the beta chain, normally the 6th amino acid is glutamic acid. But in sickle cell anemia, this glutamic acid is replaced by valine. Okay, now let's see why this happens. Each amino acid, each of these amino acids, okay, is coded by a sequence of 3 nucleotides on mRNA. So each of these amino acids is coded by a sequence of 3 nucleotides on mRNA. Okay. Here, glutamic acid is coded by GAG. Glutamic acid is coded by GAG. But in sickle cell anemia, this GAG is replaced by GUG. Okay. But GUG codes for valine. GAG codes for glutamic acid, whereas GUG codes for valine. So, in sickle cell hemoglobin, on the 6th position of beta hemoglobin chain, this glutamic acid is replaced by valine because there is a change in the sequence on mRNA from GAG to GUG. Now, this happened, okay, this change in nucleotide, okay, this replacement of nucleotide, U replaces A on mRNA sequence. This happened due to point mutation in the hemoglobin gene. Point mutation in the hemoglobin gene. Now, point mutation is nothing but substitution or replacement replacement of a single nucleotide on DNA, of a single nucleotide in DNA. In normal hemoglobin gene, the sequence for glutamic acid in the coding strand is GAG. So this is a normal hemoglobin gene. Okay, here you can see that the sequence for glutamic acid in the coding strand is GAG, whereas on the template strand, which is complementary to the coding strand, it is CTC. The sequence on mRNA is complementary to the sequence of template strand. So this sequence of mRNA is complementary to the sequence of the template strand here. Okay, and that is nothing but GAG. That is nothing but GAG. Here CTC is the template strand. So GAG is the complementary, okay, sequence on mRNA for this template sequence CTC. But in sickle cell, here you can see, in sickle cell hemoglobin gene, okay, T replaces A. So normally there is GAG in the coding strand. So in the place of A in the coding strand, there comes T. So T replaces A, or A is replaced by T. So instead of GAG, now it becomes GTG. Okay. So T replaces A. So there should be A here. Instead, there is a T in the sickle cell hemoglobin gene, that is HbS gene, which is abnormal. Okay. So we can say A is substituted by B in the coding strand. Okay. Then what happens? The template strand, in the template strand, okay, in the place of T, now there is A. Here you can see there is T here, but here it becomes A, because in the coding strand A is replaced by T. So as a result, in the template strand, okay, which is complementary to this coding strand, okay, there comes A. Now because of this, mRNA sequence also changes, that is GUG. So sickle cell anemia is an example, okay, it is an example for A to T mutation. A to T mutation, because A is replaced on the coding strand by T. It is also called AT to TA mutation. It is called AT to TA mutation. Thalassemia. Now, thalassemia is also blood-related recessive disorder. It is characterized by reduced rate of synthesis of one of the globin chains. So there are two globin chains in two types of globin chains in hemoglobin. Two alpha chains are there, two beta chains are there that make up the hemoglobin. If the synthesis of either of the chains or both the chains is reduced, then it causes thalassemia. Now this defect is due to either mutation or deletion in the genes that code for different chains of hemoglobin. There are two types of thalassemia, alpha thalassemia and beta thalassemia, based on which hemoglobin chain is defective. Now first we shall discuss about alpha thalassemia. Normal alpha hemoglobin chain is coded by two pairs of genes. Here you can see. So the normal synthesis of alpha chains, okay, it is coded by, okay, I can say the normal alpha hemoglobin chain is coded by two pairs of genes, HbA1 and HbA2, which are located on chromosome number 16. Now these two are HbA1 genes, one pair. The second pair is HbA2. HbA1, one allele pair, HbA2, another allele pair. So there are two pairs of genes which code for normal synthesis of alpha chain in the hemoglobin. Okay, so these genes are located on chromosome number 16. If there is a mutation or deletion in one or more of these genes, then abnormal quantity of alpha chain is produced. okay, leading to alpha thalassemia. So, in short, alpha thalassemia is nothing but okay, defective alpha globin chain is produced. Okay, not only it is defective, it is produced in less quantity. Okay, and the genes which are responsible for alpha thalassemia, okay, or you can say the genes which are responsible for normal alpha chain synthesis, they are located on chromosome number 16. There are two pairs HbA1 and HbA2. If there is a defect or deletion in these genes, then alpha thalassemia is caused. Next is beta thalassemia. Now, the norm for normal synthesis of beta globin chain, okay, beta globin chain, there are a pair of factors, okay, a pair of genes which are located on chromosome number 11 are responsible. If there is a defect or deletion in these genes, then it will result into beta thalassemia, in which defective beta globin chain is produced. Now, what is the difference between sickle cell anemia and thalassemia? Remember, sickle cell anemia and thalassemia both are blood related, or I can say hemoglobin related recessive, autosomal recessive disorders. Okay, but the main difference is you can see both thalassemia and sickle cell anemia are autosomal linked recessive disorders. Thalassemia is a quantitative problem of synthesizing too few globin molecules, whereas sickle cell anemia is a qualitative problem of synthesizing an incorrectly functioning globin. It means in case of sickle cell anemia, the non-functional globin chain is synthesized, whereas in case of thalassemia, okay, the quantity of the hemoglobin is less. Next, phenylketonuria. So it is also autosomal linked recessive disorder. Okay, it is an example for inborn errors of metabolism. In normal metabolic pathway, this amino acid phenylalanine is converted into tyrosine. Now this reaction is catalyzed by an enzyme called phenylalanine hydroxylase. In phenylketonuria, the gene that codes for this enzyme becomes defective. As a result, the enzyme is not produced and phenylalanine is not converted into tyrosine. Instead, this phenylalanine it is converted into phenylpyruvic acid. Now, this phenylpyruvic acid is a toxic compound which accumulates in the brain and damages the nervous system and causes mental retardation. So all these three diseases, sickle cell anemia, thalassemia and phenylketonuria, these are autosomal linked recessive disorders. Allosomal linked recessive disorders have been already dealt with. So we are not going to discuss that here. Now let's discuss chromosomal disorders. Here we should know certain terms. Now euploidy. Euploidy refers to the presence of normal chromosome number. Presence of normal chromosome number. So in humans there are 44 autosomes, that is 22 pairs of autosomes, 44 autosomes and a pair of sex chromosomes. In males, the sex chromosomes are X and Y. In case of females, there are two X chromosomes. There are two X chromosomes. Aneuploidy. It refers to change in the number of normals, change in the number, okay, of normal set of chromosomes. It results in chromosomal disorders like Down syndrome, okay, Down syndrome, Klinefelter syndrome, and Turner syndrome, etc. The aneuploidy is of two types, monosomy and trisomy. Now, monosomy refers to loss of one of the chromosomes from a pair of chromosomes. Okay, so there are different pairs, 23 pairs of chromosomes are there, right? So out of these 23 pairs, if one pair has, okay, lost, okay, from one pair, one chromosome is lost, then we use the word monosomy. For example, Turner syndrome. For example, Turner syndrome. There should be two X chromosomes here, but there is only one X chromosome in Turner syndrome. Now, trisomy refers to presence of an additional chromosome in a pair of homologous chromosomes. Okay, so here one chromosome is missing in one of the pairs of chromosomes. Here, one chromosome is there is an additional copy of a chromosome. Okay, in one of the pairs of chromosomes. Example, Down syndrome. So in trisomy, the number of chromosome will become 47 total number. In monosomy, it will become 45. Okay. So example for trisomy are Down syndrome and Klinefelter syndrome, because in Down syndrome there are total 47 chromosomes, 45 autosomes plus 2 allosomes, and in case of Klinefelter syndrome, 44 autosomes plus 3 allosomes, 2 X chromosomes and 1 Y chromosome. Now let's see different chromosomal disorders in detail. First one, Down syndrome. Now, this Down syndrome is caused due to the presence of an additional copy of the chromosome in chromosome number 21. So, that is 21st pair. In the 21st pair, there is an additional copy of chromosome. That is what is called trisomy of 21. Now, this is the karyotype of the Down syndrome. Here you can see in 21st pair, there should be only two chromosomes, but there is an additional extra chromosome is here. Okay. Now, this is the karyotype of Down syndrome. So the chromosome complement or karyotype of Down syndrome is 45 autosomes, because there is one additional copy here, plus XX or XY. It means Down syndrome may occur in males as well as in case of females. Symptoms of Down syndrome. Now here the affected individual is short statured, means he is extremely short, with small round head, here you can see, and furrowed tongue. He has a furrowed tongue, here you can see. The tongue is furrowed and partially open mouth. Palm is generally broad with characteristic palm crease. Physical, psychomotor, and mental development is retarded. So generally, the people who are suffering from Down syndrome, it is commonly occurs in children. Okay, so they are mentally retarded. Next, Klinefelter syndrome. Now, Klinefelter syndrome is caused due to the presence of an additional copy of X chromosome. Okay, so generally Klinefelter syndrome, the person who is suffering from Klinefelter syndromes, they are sterile males. So the normally there will be one X chromosome and one Y chromosome in a normal male. But here, in case of Klinefelter syndrome, there is an additional copy of X. Here you can see. So hence making it XXY instead of XY. There is an additional X chromosome, making it XXY. Okay. Now, chromosomal complement of Klinefelter syndrome or karyotype of Klinefelter syndrome is 44A, means there is no change in autosomes. 44 autosomes are there, but there is an additional X, X chromosome. So 44A plus XXY. Now, the symptoms of Klinefelter syndrome. As we have already discussed, that Klinefelter syndrome, okay, the persons who are suffering from Klinefelter syndrome, they are sterile males. They have a tall stature, so they are extremely tall. Okay, means they are abnormally tall. Here you can say, and they show a special feature called gynecomastia. So gynecomastia is nothing but breast development. So normally breast development occurs in case of females, but here breast development occurs in case of males also. That is why the name is used, gynecomastia. And since they are sterile, they have underdeveloped testes. Testes are not properly developed. Next, Turner syndrome. It is caused due to the absence of one of the X chromosomes. It's caused due to absence of one of the X chromosomes. So the persons who are suffering from Turner syndrome, they are sterile females. So normally in females there are two X chromosomes. There is no change in the allosomes, number of allosomes. But okay, here in Turner syndrome, only one X chromosome is present instead of two. So normally in case of females, there are two X chromosomes. So this second X chromosome is lost in the person suffering from Turner syndrome. So there is only one X chromosome. So chromosomal complement of Turner syndrome or karyotype of Turner syndrome is 44 autosomes plus X. Instead of 2X, there is only 1X. So like this, in Down syndrome, the number of autosomes changes. But in Turner syndrome and Klinefelter syndrome, the number of allosomes changes. Now, symptoms of Turner syndrome. So the persons who are suffering from Turner syndrome, they are short-statured compared to, it is contradictory to Klinefelter syndrome. So normally Klinefelter syndrome persons, okay, they are extremely tall. They are tall statured, whereas the persons who are suffering from Turner syndrome, they are short statured and they have a webbed neck. They have a webbed neck, and also they have underdeveloped breasts. Okay, it should be normally developed in case of females, and they also have rudimentary ovaries or non-functioning ovaries, and there is no menstrual cycle exhibited by these women who suffer from Turner's syndrome. Now let's solve some problems. Question number 1. Woman with 47 chromosomes due to 3 copies of chromosome number 21 is characterized by. Now an additional copy of the chromosome in 21st pair of chromosomes is observed in case of Down syndrome. Okay, it may occur both in males as well as females. So here answer is Down syndrome. So in Down syndrome, in the 21st pair of chromosomes, there is an additional chromosome that leads to Down syndrome. Sickle cell anemia is. Sickle cell anemia, phenylketonuria, thalassemia, all these are autosomal recessive disorders. They are all autosomal recessive disorders. An example for autosomal dominant inheritance, okay, we already studied, that is myotonic dystrophy. For X-linked recessive inheritance, examples are thalassemia and color blindness. For X-linked dominant inheritance, the example is Rett's syndrome. Question number 3. Klinefelter's syndrome is caused due to, here answer is 2X and 1 Y chromosome. Now, Klinefelter syndrome occurs in case of males. Normally, males have one X chromosome and one Y chromosome. But in Klinefelter syndrome, there is an additional copy of X chromosome. That is why there are two X chromosomes and one Y chromosome. That is XXY. So here answer is this. Now, if there is only one X chromosome, then it is Turner syndrome. Point mutation involves insertion, duplication, deletion. Insertion means there is an extra nucleotide in the normal sequence of DNA. Duplication means the same nucleotides becomes double. Okay, there are more than one copies of the same nucleotide in the sequence. Deletion means the deletion of that nucleotide. Here point mutation involves change in the single base pair. Okay. So point mutation involves change in single base pair, as you saw in case of sickle cell anemia. So single base pair change in DNA caused change in the codon present on mRNA. As a result, incorrect amino acid is produced in beta chain of hemoglobin. Gynecomastia. So this we have discussed here. Gynecomastia is nothing but the development of breasts in case of a person suffering from Klinefelter's syndrome. So here the answer is gynecomastia is a common feature seen in Turner syndrome. Oh, so in the options, these are incorrect options. The actual answer here is Klinefelter's syndrome, not Down syndrome, not Turner syndrome. So answer should be Klinefelter syndrome, which is not mentioned here. So please note that gynecomastia is nothing but the development of breasts in the person suffering from Klinefelter syndrome.